Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, so we've now had an in-depth study of the voltage-gated potassium channels, we've studied the calcium-gated potassium channels, we've studied the voltage-gated sodium channels. Now what we're going to move on to is the voltage-gated calcium channels, which for short I will be abbreviating down to VGCCs, okay, for voltage-gated calcium channels. So we're going to start off by discussing the structure of voltage-gated calcium channels. Okay, so just like in the case of the voltage-gated sodium channels, where there was one subunit to rule them all, which was the alpha subunit of the voltage-gated sodium channels, in the voltage-gated calcium channels, again, there is a principal subunit, which is the one that, if you have just this subunit, you have a functional voltage-gated calcium channel, and you don't need anything else attached onto that, basically, to have a functional voltage-gated calcium channel. Okay, we're going to start off by studying that subunit, and then we'll look at the accessory subunits. And accessory subunits are a big thing in voltage-gated calcium channels, as we'll see later on. Okay, so let me firstly then just draw you a crude picture of this fundamental principal subunit of the voltage-gated calcium channel, which forms the actual pore portion. Okay, so here it is, sitting in the cell membrane here. Here's the pore down the middle, and this is analogous to the alpha subunit of voltage-gated sodium channels. However, it's not merely called the alpha subunit of voltage-gated calcium channels. Instead, we're going to make it slightly more fancy. We're going to call it the alpha 1 subunit of voltage-gated calcium channels. Okay, and we'll see later on that there is an alpha 2 portion as well. Okay, which is part of an accessory subunit, but we'll come on to that later on. Okay, so alpha 1 subunits of voltage-gated calcium channels, these are the principal subunits. These actually form the pore through which calcium can pass through the membrane. Okay, and of course it will usually be passing in from the extracellular fluid into the cytoplasm of the cell. Okay, now alpha 1 subunits of voltage-gated calcium channels, they are a single protein, okay, not a tetramer, just like the alpha subunit of voltage-gated sodium channels. In fact, incredibly like the alpha subunit of a voltage-gated sodium channel. And in fact, if you want me to draw out the picture of an alpha 1 subunit of a voltage-gated calcium channel, I don't need to, okay, because the picture is exactly the same as what we drew for voltage-gated sodium channels. I would just be drawing out the same picture again, basically. Okay, this is the membrane-spanning topology of an alpha 1 subunit of a voltage-gated calcium channel, basically. It has these 4 domains, domain 1 to 4, which are analogous each to an alpha subunit of a voltage-gated potassium channel. Okay, they're linked together by these linkers between the domains, 1,2 linker, 2,3 linker, and 3,4 linker. And the thing folds up into this same structural arrangement here with domain 1, then followed by domain 2 here, domain 3 here, here's the voltage sensing domain, here's the pore forming domain, and domain 4 here. Okay, so everything's very much so analogous. Okay, right, so that's the structure then of these alpha 1 subunits of voltage-gated calcium channels. Very, very analogous to the alpha subunits of voltage-gated sodium channels. Let's now discuss the diversity of different alpha 1 subunits of voltage-gated calcium channels. Because we saw in the case of the alpha subunits of voltage-gated sodium channels that there wasn't just one alpha subunit of voltage-gated sodium channels. There were all those 9, remember. NAV 1.1, NAV 1.2, all the way up to NAV 1.9. Well, again, in the case of voltage-gated calcium channels, there are 10 genes for alpha 1 subunits of voltage-gated calcium channels. Okay, and these 10 genes are going to be classified into 3 families, basically. Okay, so let me firstly show you the terminology, or rather the notation. Okay, so we put Ca for calcium, then we put a subscript for voltage-gated, so voltage-gated calcium channel. Okay, and then the first family is going to be called the CAV1 family. Okay, the second family is then going to be called the CAV2 family, and the third family is going to be called the CAV3 family. Okay, and the CAV1 family is going to contain 4 genes, which will be called CAV1.1 all the way through to CAV1.4. So there's CAV1.1, CAV1.2, CAV1.3, and then finally CAV1.4. And those are these 4 genes for alpha-1 subunits of voltage-gated calcium channels that are in this CAV1 family. And the CAV2 family, that contains 3 genes for alpha-1 subunits of voltage-gated calcium channels. Okay, and these are called the CAV2.1, the CAV2.2, and then the CAV2.3 gene. Okay, so those are the names for these 3 genes for alpha-1 subunits that are classified into this CAV2 family. Okay. Finally, the CAV3 family, that must also contain 3 members to take us up to the total of 10 genes, and indeed it does, CAV3.1, CAV3.2, and then finally CAV3.3 here. Okay, and I'll underline that family in blue. Okay, right, so there are the 10 genes for alpha-1 subunits of voltage-gated calcium channels arranged into these 3 families. Okay, now, voltage-gated calcium channels, as I said at the start, they're going to have lots of accessory subunits usually attached onto these alpha-1 subunits. Very rarely do you ever actually have a voltage-gated calcium channel that only consists of the alpha-1 subunit. Okay, however, the alpha-1 subunit is the most important subunit of the voltage-gated calcium channel for deciding the properties of the voltage-gated calcium channel. What do I mean by properties? I mean things like what sort of voltages it activates at, how long it's open for after it's actually been activated to open, things like that. Okay, so, and it's the alpha-1 subunit that is major in deciding those things. The accessory subunits contribute, but the alpha-1 is absolutely major, and for that reason, voltage-gated calcium channels are named according to which alpha-1 subunit you actually use to produce them, basically. Okay, so I'm now going to tell you about the different naming systems. So basically, if you use one of these genes in the CAV1 family to produce your alpha-1 subunit that you're going to use in your voltage-gated calcium channel, then your voltage-gated calcium channel will be what's known as an L-type voltage-gated calcium channel. Okay, so when people talk about L-type voltage-gated calcium channels, what that actually means is that this voltage-gated calcium channel they're referring to, its alpha-1 subunit is an alpha-1 subunit that is coded for by one of these 4 genes here. Okay, so either this alpha-1 subunit is a CAV1.1 type alpha-1 subunit, or it's CAV1.2 or CAV1.3 or CAV1.4. That's what is meant when people say an L-type voltage-gated calcium channel. Okay, right. Now let me just talk a little bit more about L-type voltage-gated calcium channels. Let me give you some examples of where they are very important. So in skeletal and cardiac muscle cells, L-type voltage-gated calcium channels play an incredibly important role in the excitation-contraction coupling. Okay, so the coupling between having an action potential propagating along the cell membrane of the skeletal or cardiac muscle cell, and then the actual calcium signal generated inside the skeletal or cardiac muscle cell, which then causes the contraction of that cell. Okay, and the L-type voltage-gated calcium channels that you find in skeletal muscle, okay, are actually specifically made out of alpha-1 subunits that are produced from the CAV1.1 gene. So specifically in skeletal muscle, you have L-type voltage-gated calcium channels that use this CAV1.1 type alpha-1 subunit. Okay, so that's the one found in skeletal muscle. Okay, let me now talk about the one that's found in cardiac muscle. The one that's found in cardiac muscle is CAV1.2. So the L-type voltage-gated calcium channels that you have in cardiac muscle cells have alpha-1 subunits that are from this CAV1.2 gene, basically. Okay, right. So those are some notable examples of where these CAV1 genes are actually used. CAV1.1 is skeletal muscle, and CAV1.2 is used in cardiac muscle. Okay, but L-type voltage-gated calcium channel, all that tells you is that the alpha-1 subunit is one of these types in this CAV1 family. Okay, now, there are other types of voltage-gated calcium channel that people refer to. For instance, things like N-type voltage-gated calcium channel, PQ-type voltage-gated calcium channel, R-type voltage-gated calcium channel. Okay, and those concern voltage-gated calcium channels that have certain types of CAV2 alpha-1 subunits. So basically, if you build a voltage-gated calcium channel where the alpha-1 subunit is a CAV2.1 type alpha-1 subunit, then the voltage-gated calcium channel you end up with is called a PQ type voltage-gated calcium channel. So PQ-type voltage-gated calcium channel is more specific than L-type voltage-gated calcium channel. L-type just tells you that the alpha-1 subunit is from one of these CAV1s. Okay, PQ-type tells you the exact gene in the CAV2 family which coded for the alpha-1 subunit in that voltage-gated calcium channel. Okay, similarly, CAV2.2, if you use CAV2.2 in your voltage-gated calcium channel, then that voltage-gated calcium channel will be called an N-type voltage-gated calcium channel. Okay, so I'll just underline that one in red. So when people talk about N-type voltage-gated calcium channels, that's what they mean. They mean voltage-gated calcium channels where the alpha-1 subunit is CAV2.2. And PQ-type and N-type voltage-gated calcium channels are really important in the axon terminals of neurons. They are responsible for turning the electrical signal, the action potential arriving in the axon terminal, into a calcium signal, calcium coming into the cytoplasm of the axon terminal, which will then trigger neurotransmitter exocytosis from the axon terminal. Okay, next up, finally, I want to talk about R-type. Okay, so if you use the CAV2.3 type alpha-1 subunit to build your voltage-gated calcium channel, you end up with what's called an R-type voltage-gated calcium channel. Okay, so if you hear people talking about R-type voltage-gated calcium channels, it just means a voltage-gated calcium channel where the alpha-1 subunit is specifically a CAV2.3 type alpha-1 subunit. Okay, so the different genes in the CAV2 family, the voltage-gated calcium channels they give rise to are all given these different names. Now, when we go back into the CAV3 family, it goes back to being much broader, like it was for L-type voltage-gated calcium channels. Okay, so if you make a voltage-gated calcium channel which has as its alpha-1 subunit one of these three types, a CAV3.1, a CAV3.2, or a CAV3.3 type alpha-1 subunit, then your voltage-gated calcium channel is called a T-type voltage-gated calcium channel, or a T-type VGCC. Okay, so again, that's much broader. It doesn't tell you specifically what gene the alpha-1 subunit was coded by. It tells you that it was coded by one of these three in the CAV3 family. Okay, and some more information then about the CAV3 family is that the voltage-gated calcium channels that are important in the action potential within the sinoatrial node are T-type voltage-gated calcium channels, and those T-type voltage-gated calcium channels in the sinoatrial node of the heart are ones which consist of CAV3.1 or CAV3.2 alpha-1 subunits, basically. So these genes code for the alpha-1 subunits of the voltage-gated calcium channels that are really important in the action potential in sinoatrial node cells. So remember, in the sinoatrial node of the heart, the action potential that occurs there is different from the action potential that occurs in other cardiomyocytes. Okay, and the upstroke of the action potential there is not triggered by voltage-gated sodium channels, but instead by T-type voltage-gated calcium channels. And I'm now telling you that those those T-type voltage-gated calcium channels that are important in the sine-radial node are specifically T-type voltage-gated calcium channels where the alpha-1 subunit is either CAV 3.1 or CAV 3.2 type. Okay, right. Now I'm afraid I'm going to have to actually cut it there for this video. Okay, we will continue this discussion in the next video.